Firstly, I guess the title of the talk is building or breeding better soils. And here's that famous triangle that you spoke about. And because I'm a bit of an egomaniac, obviously I put biology at the top, but it doesn't have to be that way. And as you so rightly mentioned, when you start to look at these things, so when I'm talking about biology, I'm talking about your microbes, your biomass, your bioavailability of nutrients, which Edward also spoke about. And then down here you have your structure and your chemistry. And as Edward so rightly mentioned, these things are all interdependent. So don't take them as independent factors. As you change one, the others will change. So I guess because it's World Soil Day by tomorrow, I should probably point out a fact that a lot of people probably don't know. We lose 10 million hectares of arable land every year due to erosion. Okay? So we're talking about arable land, we're not even talking about land in total. If you start to think about that, that's about 1.5 times the size of Tasmania. Every year gets lost. And it has a lot to do with our poor soil management throughout the world. So not just in Australia, we're talking about in other places, in Africa, in Asia, in South America, even in some parts of Europe where we're just not managing our soils well. So I just want you to be aware that, we do need to look after what we've got. So I'm really going to be talking about two of the main factors, or two of the main organisms in the soil, bacteria and fungi. Now, when you take a teaspoon or say a cup of soil, that can have up to 30,000 different species of bacteria and fungi in it, okay? A teaspoon of soil will have up to 1 billion microbes in it. So if you think about taking a cup of soil, I'm talking about active soil, so I'm talking about soils, for example, in a forestry setting, where there's lots and lots of organic matter in that soil. In that single cup, there's more microbes in there than all the people that have ever lived on this planet and all the people that are currently living on it, okay? So we're talking about huge numbers. Now, there's enormous variability. Again, Edward touched on this. And there's obviously different soil types, and there's also different plants that are growing within those soils. Some areas may be dominated by fungi. So if you start to look at a woodlot, or you're looking at a forestry, lots and lots of fungi there. And, obviously when you go walking through woods, you'll see these fungi as these mushrooms that could pop up. In other areas, so in grassland areas, tends to be dominated by bacteria. And obviously there's huge variations within seasons. So every spring versus every summer versus every winter, there's all this variability. So the number of microbes and the diversity varies all the time. Now, most microbes are really poorly described. So we've looked at some of these microbes where I work, and we get them identified. And I kid you not, I go and do a Google search, and what I hear or what I get back sometimes is, this has been isolated from the bum of a cow. I mean, literally they go, this is a rumen microbe. We know nothing. We don't know what it does. We don't know any other functions that it potentially does. So, they're very poorly described. We can only grow about 5% in the laboratory, and so we don't know what many of them do as yet. But this is what we do know. The benefits of soil microbes are enormous for both plant growth and health. So we're talking about nutrient acquisition. So I'll talk a little bit about that issue with phosphate solubility or insolubility as maybe the part thing. Breaking down organic matter. So, degrading cellulose. And I'll be talking about CO2 as a plant nutrient. Nutrient manufacture. So this capturing of nitrogen that is so critical. So when we think about nitrogen and the use of nitrogen, 70% of all the nitrogen that is used in agriculture is actually due to microbes, bacteria in this case, capturing nitrogen from the atmosphere. The remaining 30% comes from inputs of fertilizers. Disease protection, really, really important, and what microbes do there. And microbes, surprisingly, also produce plant growth hormones, including auxins, which are the sort of things that you find maybe in rooting powders and rooting gels. They stimulate root development, and cytokinins tends to stimulate plant growth at the tips. So this is a little bit of science, and I'm trying not to be too heavy on the science aspect, but a lot of people seem to be of this impression that the plants in that plants just sit there and are really inactive or very laid back. They don't do much. But plants are actually really, really active in how they select the microbes they want to live with. So let's take an example. the ability to form nodules that you see on legumes. So if you ever pulled out a legume, whether it's a pea plant or some clover in your lawn, you'll see these little nodules on the roots. So what happens is the bacteria produce something called a nod factor, and that basically just tells the plant that this type of bacteria is present. And at the same time, the plant secretes flavonoids. So that's sort of like a signal that it secretes into the soil to recruit these microbes. It goes, come here, I need nitrogen, I need you to help me fix nitrogen. I'm sending you a signal to come, and the bacteria responds by saying, hey, here's a signal, I'm here. And when those two things happen, the root hair curls around, and eventually you end up with this nodule, okay? So bear that in mind, that's absolutely critical. Similarly, you're probably aware that a lot of plants form really, really tight relationship with a whole range of fungi, okay? And that's really, really critical for plants, especially in nutrient acquisition, but there's a whole range of other things, including disease suppression and the like. Now, if you're really interested in that, I suggest you have a look at the Wood Wide Web. It's basically all about fungi and how they interact with plants. But very similar things happen. In this case, the plant again secretes certain factors into the soil. The fungi signals with what's called a MIC factor for a mycorrhizal factor. And again, that then allows that fungi to interact with the roots. Because the plant doesn't necessarily want to interact with the fungi if it's going to be a pathogen. Okay, so it's looking again for a signal. I'm sending a signal because I need a fungi, and the fungi sends a signal back saying, I'm here, please allow me to interact with you, versus I'm not sending you a signal and therefore I'm going to be a disease, potentially a disease-causing pathogen or a disease-causing fungi. So let's have a look at this fixation of nitrogen. So what happens is you get these nodules on the roots, okay? And generally the species for legumes are rhizobiums, but this is not restricted to just the legumes that we know of. A lot of other plants do it as well. For example, your Allocasuarinas and your acacias also actually do this type of thing. They also interact with these microbes. So they form these nodules. When you cut these nodules open, you realize they're full of bacteria, okay? And so they're busy fixing nitrogen. Now, here you'll see a peanut plant, and this is uninoculated. So this hasn't been given the microbe that can fix nitrogen. This is the same plant if it's given the microbe that can fix nitrogen. So there's absolutely no doubt there's a huge difference. Now, exactly the same experiment was done, but what they did is they took that microbe and they knocked out that nod factor. Remember I told you that nod factor, that's so important for the bacteria to signal the plant and say, hey, here I am, let's interact. You knock that out, and basically the plant will not accept that microbe and it will not fix nitrogen. Okay, so you can see how critical that interaction is. Now, obviously not everybody is going to grow legumes and the like, but there's a whole range of free-living microbes as well that also fix nitrogen in the soil. Perhaps the best known is Azotobacter and some of the things like the Pseudomonads, but the one that you guys are probably familiar with is what I call cyanobacteria. I hate the term blue-green algae because it's not an algae, but that's probably the term that a lot of people know. So when you go, on the Torrens on a nice sunny day and it's been warm, we've just had a rain shower, and all of a sudden you get there and you realise the entire Torrens has turned this beautiful shade of jade, right? That's all those cyanobacteria. What the cyanobacteria do is, well, because they can fix nitrogen, but they can also fix carbon, so they can make their own food source, all they need is a bit of material from the excess use of fertiliser flushed into the Torrens, bang, and you'll get this algal bloom, okay? So they're very, very good at doing what they're supposed to do. And obviously, if you do encourage these types of microbes in your soil, you can fix about 30 to 40 kilos of nitrogen per hectare. That equates to, well, a tonne of urea probably has about 600 kilos, so it's probably about 50 kilos of urea per hectare. Now, Edward was talking about how phosphate gets locked up. Potassium can also be locked up. And really the reason it gets locked up is that phosphate has a negative charge on it, okay? And so anything it encounters in the soil that's got a positive charge, let's say calcium or aluminium or iron, it immediately binds and forms this molecule that can't be taken up by plants. So what happens is these bacteria, they make that phosphate soluble. So you see these clear zones around the colony. This is insoluble phosphate, the white material. That's soluble. So when you have those, that phosphate becomes soluble, the plants can take it up, okay? So again, it's really critical that this type of system is working in your soil, because what it means is that not only can you unlock existing phosphate, but you actually can increase your fertilizer use efficiency. So you can get away with putting the same amount or less phosphate on, but get the same or even better result for it. Similarly, what we do at Neutrog is we also look for other microbes. So here you'll see one that can unlock potassium. So potassium can also be locked up. Over here you'll see on these plates that these microbes here have the ability to degrade cellulose. So when you put organic material down, we need to start to break that down. And one of the ways we do that is by actually being able to digest cellulose. One of the other things that we look at is the ability to produce plant growth hormone. So in this case, what we do is we grow all the individual bacterial species up, we remove the cells, and then we basically measure to see how much plant growth hormones gets produced. And we've got some strains that produce enormous amounts of plant growth hormone. So when you start to put these together into your soil, what it means is that you get better seedling establishment, and you also get your early plantings and your seed germination is accelerated. So again, it's another function that soil microbes do. So, when we're looking at this, we're looking at things like the auxins, the cytokinins, and the gibberellic acid. So auxins, like I said before, it's found in rooting powders and rooting gels. You also see some cytokinins. And gibberellic acids, they actually change the internodal length, so how tall does a plant grow between where the leaves come out. Gibberellic acid is actually used in places like Hawaii to increase the internode length in sugarcane, because they can basically end up with more sugar being in that thing. So, these things are produced naturally by soil bacteria, and they're actually used as a signaling molecule to talk to one another. But they're also produced by plants. Cytokinin is a similar sort of thing. As I mentioned before, the cytokinin stimulates these cells to divide and to grow, and therefore you get better plant growth. And the gibberellic acids were actually first found in a fungi in rice, and the rice was growing so tall it just kept falling over. Okay, so these are all produced by microbes, but also produced by plants. Now, the interesting thing is that auxins are produced in the tips of the plant, yet they're required at the roots. And the gibberellins tend to be produced at the bottom of the plant, but are required at the tip. I don't know who invented this system. Wouldn't have been me, but hey. But if you think about it, as Edward pointed out, everything moves only if there's water. So the cytokinins move quite readily up the plant, because obviously as water carries it along, it goes along in the xylem, sorry. But the auxins actually have to be transported down. That actually takes energy, all right? So if you can produce them down here by soil microbes, you're doing the plants a favour. Obviously, these hormones all work in combination. So when you think about hormones in people or in animals, it's always a balancing act. So when you have a lot of auxins, you get root formation. If you have a high cytokinin count, you get the development of the shoots. And when you have about a 1-to-1, you get healing of the plant. So when that plant forms that callus where you've cut the branch off, that's all due to a regulation of hormones telling the plant this is what you need to do to heal that wound. Okay? I want to move on to fungi now as the sort of second pillar of what we want to talk about. So the fungal plant symbiosis is really interesting, and there's 90 or 80% of plants probably form this, what we call an endomycorrhizal interaction. So endo just means internal, like we've got an endoskeleton, and we have what's called an ectomycorrhizal interaction, which generally occurs with woody plants. So ecto just means external. So, when you step on that cockroach and it makes a sound, that's just the ectoskeleton breaking. So, because they have a skeleton on the outside. So the difference really isn't that great, except that the invasion or the depth into which the fungi goes and the root isn't as great as it is for these endomycorrhizal fungi. But why would a plant bother to do this? I mean, come on. Why do I need a fungi? Well, if you believe it or not, plants secrete about 30% to 40% of all the sugars they produce in their leaves back into the ground. And that's to encourage these bacteria and fungi. And here's the reason why. You have an area through here, we call that the depletion zone. So you see the root comes down and you've got these root hairs. Once the plant has taken the nutrients out of that area, there's really no way of getting many of those nutrients. Some nutrients will move. So if you're thinking about things like sulfates and nitrates, they move quite readily in the soil. But others like phosphate and potassium, they don't really move within the soil profile. What happens is you get this interaction. The fungi sends out these hyphae. The fungi is solubilizing that phosphate for the plant to be able to take it up. So we're talking a 1,000-fold increase in ability to access certain nutrients if you have these. Similarly, if you look at this little plant here, you realize that the majority of what you see through here is actually that hyphal network. It's not the root system of the plant. Okay, so you can see how important it is. It increases the surface area enormously. A plant without this is maybe in contact with 3 to 4% of the soil particles. So obviously with this system, the plant is in fact in contact with a much larger number of soil particles and has a much better way of acquiring nutrients. One of the other things that fungi are really good at is controlling and killing nematodes. So hopefully, would you be so kind of, yep, thank you. So you see the little nematodes wriggling around here? There's some eggs up here. This one is about to pop some eggs out. What you see through here is that fungal network, okay? Over time, that fungus digests these nematodes. And the reason it does it is to a fungi, those nematodes represent a little bag of protein. And what's protein? A source of nitrogen. See, it's being digested away. It's a really, really great system for controlling nematodes. However, if you put a lot of nitrogen fertilizer on the soil, the fungi don't do this. Because it's a bit like me. Why would I cook if somebody's delivering me Uber Eats every day? Okay? Exactly the same thing. I don't do it. I'm lazy. The fungi is lazy. It's going to do whatever is most energetically favorable. So use a lot of nitrogen-rich fertilizers. This system won't exist in your soil. Okay? Now, I want to move on to CO2, and Edward touched really well onto CO2 and carbon. And normally my talk goes about an hour and a half, but I've tried to shrink it down a little bit. So when you have a look at plant growth relative to CO2 concentrations, we're somewhere around this part here, so about 400 parts per million. You can see some plants respond really well to increases in CO2, others less so. But when you put your mulch out or you're putting out your compost, 80 grams for every 100 grams you put out disappears again as CO2. Again, the point that Edward made was that you can't just build soil carbon by throwing lots of stuff out and hoping within 6 months it's all going to be hunky-dory. It isn't. Some gets used by microorganisms to grow. Some of it becomes humic compounds. So you're probably only looking at about 10% retention, okay? But there is something really interesting that happens when you increase CO2 levels. The stomatal pores at the base of the leaves close when the CO2 concentration is higher. So when you're mulching or when you're putting out compost, what's happening is the microbes are breaking that material down. You get localized increase of CO2. So we're not talking about global increases, we're talking about localized around your plants. When that occurs, these pores at the base of the leaves close because the plant doesn't have to open them so wide to get CO2. Plant needs to get CO2, right? And it's sort of like us, if we're breathing nice and gently, we don't get dehydrated the same degrees if we're going, sorry, it's not one of those, 1/300 lines, but basically what happens is that the plants close these pores. And what you'll find is that the water use efficiency goes up. It actually means you need to use less water to grow your plants. Now, we know mulches and compost help to conserve soil moisture, but it also actually reduces the requirement of your plants. And these experiments have been around for a long time. In the 60s, people first realized that plants were growing much better early in the morning than they were later in the day, and it has to do with their localized CO2 around the plant. it's like we're reinventing the wheel, but it's a probably discussion for another day. So now I want to move on a little bit to looking at pathogens and pathogen suppression. So this is just a plate with a pathogen growing all over it. So that's a fungi. And so we spend a lot of time looking at trying to suppress pathogens. So we screen many of our soil types for it. And there's probably several hundred different pathogens that plants are affected by. So we've chosen a few that are more likely to be relevant for the majority of people. So things like Fusarium and Rhizoctonia and these types of things. A lot of people would probably be familiar with Pythium and Phytophthora because it's a big problem at the moment. We've had so much moisture. So these are not actual fungi. They're oomycetes, but let's not split hairs for the purpose of this talk. So what we do is we grow these up, and then what we do is we start to screen to look for how can we find something that might suppress a fungi. So when we get soil samples in, we basically dilute them, we plate them onto various plates. So you'll see all the different types of bacteria and fungi there. Sometimes you get lucky and you see a nice zone around a colony, and you know that one, in this case here, that's one that solubilizes potassium, very similar to what I showed you before. And the idea is that, what we're trying to do is we're trying to build up a bank of these microbes. And, occasionally you get really lucky. You screen something, you immediately see this fungi growing through here, but it's not growing around this colony. And similarly here and here. So we know that those guys have the ability to inhibit a pathogen, okay? So we really do that. And sometimes you have to screen many examples to find some. So, but you'll see here, I don't know how well you can see that. I'll go to the next slide perhaps, but we have to screen lots and lots of samples, and occasionally you'll find some that inhibit. Here's a really good example. So in the middle here, we've put a pathogen, and here we've inoculated what we think are inhibitors. Now you can see the pathogen will grow quite clearly out towards there and there. It won't grow up here because that inhibitor stops it from growing. Similarly down here, it won't grow down here because again the pathogen is that the bacteria is stopping that from growing. So what I'm trying to get across is that soil microbes do such an enormous job in your garden and in your fields and wherever you are. So it doesn't really matter whether you're looking at this from a perspective as a home gardener or you're looking at this as a perspective from, say, a pastoralist or a farmer. We can all benefit by having more microbial diversity because we have all of these benefits. Now, the easiest way to do this is to start to think about what are you putting on your soil. Put on things like your composts, your mulches, your organic mulches. What that does, those things provide food for microbes. And you'll increase the numbers, you'll increase diversity over time. Okay? So really, we have this bit of an ethos. Unfortunately, this has slipped down. But diversity is really critical in order to have optimal plant health, optimal disease resistance, and optimal production. And that's why everyone's ethos should always be diversity, diversity, diversity. And I think, if we go back to the people that spoke right at the beginning, and they spoke about diversity in food and diversity in the environment, what we've done over many years here, we've degraded that. We've gone to this idea that we can do everything with chemicals, and we've neglected the life in the soil. And I think, this time of the year or this day, Soil World Health Day, is really a really good reminder that we need to look after that. I mean, if we think about what little soil there is, it's a very, very thin crust across the planet. And if you start to think about how much we have available for us to grow food, it's 1/32 of the total land mass is what we have. So you get an apple, start slicing it up, and you'll realise what 1/32 looks like. It's not a lot. And of that, it's a very thin crust. In Australia, in my own garden, I know my topsoil might be 30 centimetres. I live in the foothills. Other areas it might be a metre or so, but we've got a very, very thin crust, and we have to preserve that. We have to make the best of what we have. So I thank you for your time, and yes, I'm happy to answer questions later on in the panel.